My name is Syed, Askari Rizvi. Today, the topic of my talk is emerging peptide-drug conjugates via chemical conjugation with improved therapeutic potentials. The content of my research are my research interest, background about the peptide drugs, era of peptide drug developments, comparison of ADCs versus PDCs in terms of their structure and their mechanism of action, receptor targeting peptides and peptide drug conjugates, and finally the conclusion. So first of all, I have an excellent research experience in drug delivery and pharmacokinetics with broad spectrum focus including but not limited to various medications used in the treatment of cancer, Alzheimer's disease, and understanding the role of biopharmaceuticals and pharmacokinetics therapy. I've been author for more than 40 publications and I have given 20 international presentations on drug delivery, radiopharmaceuticals, and nuclear medicines. I have been reviewed more than 16 research articles for highly reputed journals. So first of all, the background of my talk, why we are doing work on peptides. So basically, we all know that cancer is considered as a serious life-threatening problem globally and is still the second leading cause of death worldwide. And according to IARC and WHO report, there are about 5-year median survival rate was estimated to 53.5 million among 185 countries. And till 2024, approximately 20 million cancer cases were registered. Among them, 9.7 million people died. So the most leading cancers are lung cancer, breast cancer, colorectal cancer, and prostate cancers. So medical science is indeed in the need of designing novel therapeutic drugs with reduced side effects and improved survival time. So there are a few standard treatment strategies like chemotherapy, surgery, radiotherapy, and immunotherapy, as well as peptide receptor radionuclide therapy, peptide receptor response therapy, which are using for the diagnosis and treatment of these cancers. And recently, the past era, Paul Ehrlich, a German physicist in 1910, first proposed the concept of magic bullets. What are the magic bullets? Magic bullets are the targeted drugs which can target a specific receptor or target a specific type of cancer and show its anti-cancer activity. So today, a handful of drugs were entered into clinical practice, but only a few drugs got FDA approval. We also have some kind of side effects. So to reduce these side effects, we need to conjugate the chemotherapy drugs with some targeting moieties to improve their targeting ability and their cytotoxic potential. So how peptide drug can internalize into cancer cells? There are the different routes of the internalization of these peptide drugs. And I will follow the peptide nanoparticles, peptide receptor targeted therapy, and peptide as a fluorophore carrier and peptide as a radionuclide carrier. So now the question is, can RGD tag with other moieties to improve targetability? So yes, we can use RGD tripeptide with some chemotherapeutic drugs, antimetabolites, small molecules, radionuclides, and some other moieties to improve their targeting ability, their internalization into the cancer cells and in the cancer cells. So this is the era of peptide drug developments. In 1985, the first prostate and breast cancer drugs, GnRH receptor targeting peptide is introduced, and the most recent one is 18 Posluma, which is used for prostate cancer PET imaging. Next, what is the concept of ADC? Why we are using the peptide conjugated drugs? The concept of PDCs comes from the concept of ADCs to work on the side effects of these chemotherapeutic drugs. A few decades before, growing era of antibody drug conjugates as a major class of targeted drug delivery therapeutics have emerged for the treatment of cancer. So basically, the antibody drug conjugates consist of three major parts: the antibody, which can target the specific antigen; a linker, which is serve as a bridging moiety between the antibody and chemotherapy drugs. We can use some chemotherapeutic drugs as a cytotoxic payloads. These ADCs can be served as a very good targeting and therapeutic point. So globally, a total of 14 ADCs have been approved from FDA and are currently present in the market for the treatment of various kinds of tumors. However, the therapeutic efficacy, pharmacokinetic mechanism, and issue of drug resistance are still pondering challenge for biomedical scientists. So, on the base of this concept, the adaptation of alternate targeting specific peptides in place of antibodies improves the hydrogenation nature, and enhance the homogeneity, reduce the drug reconjugation rate in circulation, and further avoid the off-target toxicity. Thus, increase the intracellular delivery of highly cytotoxic payloads. So these PDCs are composed of natural peptides, and we know that natural peptides extracted from natural components have relatively short half-life, and they can be readily metabolized by end-to-end exopeptidases. So we need to design some synthetic peptide drugs. And synthetic peptide drugs can easily be manipulated to increase in vivo stability, membrane permeability, and target specificity. So in order to detect these peptides, we can conjugate these peptides with fluorescence dyes, radionuclides, and to serve as a therapeutic moiety, we can conjugate it with some chemotherapeutic drugs. So similar to ADC, the PDC also consists of three main components. One is the targeting peptide moiety instead of antigen antibody, and the linker which serve as a bridging moiety between the peptide and chemotherapeutic drugs and the cytotoxic payload. So both have the same mechanism of action, just the difference is that the ADC target the specific antigen present on the cancer cell surface, while the PDCs target the specific receptors present on the cancer cell surface, and then they internalize into the cancer cells and lysosomal and endosomal activity intercept, and then it will cleave the linker moiety to release the drug, and then drug can show its activity. These are some chemotherapeutic drugs which have been used for the construction of ADCs and PDCs, and all these are FDA approved drugs. And similarly, we can use proton-conjugated radionuclides and nota-conjugated radionuclides. These are some chemical conjugation functionalities which can be used during the construction of linker moieties, and these linker moieties can be served as a bridging material between peptide and chemotherapeutic drugs. So it is a table of summarized all the pH sensitive linkers and sensitive linkers and redox sensitive linkers. Similarly, cleavable and non-cleavable linkers are also exist, and these linkers can easily be used for the construction of PDCs. So these are some receptors which are present on the cancer cell surface, and we can design various type of peptide moieties, and these peptides can be served as receptor targeting agents, and we can use some linker moieties like the best example of AN152, which is in clinical trials, and it is a GnRH peptide, and connected with an ester bond linker with doxorubicin. So this PDC can easily take this doxorubicin into the cancer cell, and where the linker will be cleaved and release doxorubicin. So on the basis of this concept, I designed a novel peptide, RGD-KLAK peptide moiety, and connected it with succinic acid, succinic anhydride linker moiety, and connected it with paclitaxel to improve the solubility of paclitaxel in water. And then we performed some in vitro studies, drug release studies at increasing time intervals, and different solvents like pH, PBS and pH 7.4, human blood serum, and cell culture mediums. And so we found that about 12% PDC is released in 72 hours. Remaining the paclitaxel is intact with the peptide linker moiety. Next, we studied the effect of pH and time, and at pH 5, the release of paclitaxel start gradually, and within 24 hours, almost all of the paclitaxel is completely released from PTC. Then we performed a cell viability study, and these results show that this PTC has a very excellent IC50 value, and it can easily enter into the cancer cells, as well as it can easily enter into the paclitaxel resistant cells. So the drug resistance effect is also a very important point to be considered. And paclitaxel resistant cells, the paclitaxel cannot easily enter into the cell, but when we connected it with targeting peptide, the effect is very good. Thus, we performed the confocal laser scanning microscope imaging study, and the result shows that this PTC is very well entering into the cancer cells, and in the nuclear periphery region, the drug is released. So, TAPI image and the mitotracker image shows that the peptide is entering into the mitochondria, and the paclitaxel is targeting the neuronal activities and targeted the beta tubulin 3, which is the main target species in the cell for paclitaxel, and thus they produced ROS, activated caspase 3, and thus leading the apoptosis of the cancer cells. These are some in vivo imaging studies, in vivo therapeutic studies. In vivo therapeutic studies show that the alone peptide and the alone paclitaxel do not show very much effect as compared to the peptide paclitaxel conjugate. So the tumor size is very good reduced within the 12 days, and the PTC is target to the cancer cell via alpha V beta 3 integrin receptors, which are present on the cancer cell surface. And here we can see the SPECT imaging analysis. It is the control tumor area, and here it is. We just injected the technetium 99M, and here we injected the PTC. So PTC is very well targeted the tumor tissue and accumulated into the tumor tissue, kill the tumor cells, and reduce the tumor size. So finally, with the recent advancement in the field of peptide chemistry, peptide drugs exhibit excellent stability, specifically targeting and penetrating ability, economic as well as simple and facile synthesis routes. Further, the peptide moieties provide a broader platform as a promising targeting specific agent for cytotoxic payloads and nanoparticles for an effective drug delivery system. One of the most troublesome limitations of peptide is their rapid renal clearance, and it can be easily grasped by employing multiple chemical modification techniques such as cyclization, deconformations. We can use D amino acid instead of L amino acids, chemical stapling, and nanoformulations. So thanks to the unique properties of our novel design, peptide paclitaxel conjugate, which show high water solubility and small size cyclization stability, show against the many blood serum proteins and other enzymes, as well as high ability to conjugate via covalent ester bond with the chemotherapeutic drugs. It was also observed that paclitaxel is released from PTC in its active form into the cancer cell because of chemical nature of the ester bond, disrupting the mitochondrial membrane and inducing cell apoptosis in brain tumor glioblastoma model. Thank you so much for your attention and listening carefully.